talking, so. Hello, I'm Valeria. I'd like to thank the organizers for giving me this opportunity to share our work at this meeting. I'm a graduate student in the lab of Antonio Giraldes at Yale University, and today I will tell you about a high-throughput approach that has enabled us to identify stabilizing and destabilizing sequence elements that regulate mRNA turnover during an important developmental transition. So existence in all animals begins as a single cell that has all the information to carry out embryonic development. But to enable differentiation, the cell has to first acquire a totipotent state. This reprogramming event takes place during the maternal zygotic transition, happens in all animals, and it occurs during maternal zygotic transition. This transition involves moving away from using maternally inherited mRNAs and proteins, which were used to carry out cellular functions early on, and towards activating its own zygotic genome, at which point developmental control is being passed to the zygotic genome and maternal program of development is repressed through the active clearance of maternal transcripts. We are interested in understanding mechanisms that drive this transition, and we're using zebrafish as the model organism to understand it because of several key advantages. Most importantly, because the embryo is initially transcriptionally silent, this diverse pool of maternal transcripts can only be post-transcriptionally regulated, which offers us an in vivo context to study post-transcriptional regulatory mechanisms that drive embryonic development. So it's been long appreciated that zygotic transcription triggers the clearance of maternal mRNAs, but the factors responsible for this activity have for a very long time been elusive. And the first zygotic factor known to function in this process was identified in zebrafish to be a microRNA. miR430 is a zygotically transcribed non-coding RNA that recognizes its targets through the specific sequence in the 3' UTR and targets mRNA repression and clearance. But it turns out that miR430 is not the whole story, and we've been able to detect using high-throughput mRNA sequencing, we've been able to reveal 3 distinct classes of maternal mRNAs. Using high-throughput mRNA sequencing, we defined all maternal mRNAs that are destabilized during MZT. To do that, we performed mRNA sequencing at 30-minute intervals across MZT and defined all the destabilized transcripts. And what we found is that there are 3 classes. First, if we block all zygotic transcription with an RNA-Pol2 inhibitor alpha-amanitin, we find that some maternal mRNAs are still cleared as is in wild type, while a big proportion of them are stabilized, indicating that maternal clearance involves at least 2 programs. One that requires maternal factors and one that requires zygotically transcribed factors. To distinguish the zygotic program, we can further dissect it into miR430 dependent and independent categories by blocking miR430 function. And here you see within a subset of transcripts that depend on zygotic program for clearance, the ones that are stabilized when miR430 is inhibited are miR430 targets, and a much larger group depend on an alternative, on another factor that's miR430 independent. So defining all the mRNAs that are destabilized in miR430 independent fashion has opened the door to characterizing novel pathways of mRNA regulation during development. So we don't know how this pathway works, but what we do know is that miR430 functions by recognizing specific 3' UTR sequences in the 3' UTR, and we hypothesize that this alternative mechanism also uses specific sequence signatures to recognize its targets. But of course, because there are over 1,000 of these transcripts, we can't exclude the possibility that there is more than one cis-regulatory element. And so the big question became is how do we identify how such a large number of transcripts are regulated? And to do that, we developed a high throughput strategy to look specifically in the 3' UTRs and search for destabilizing sequences that exist. To do that, we've picked out our top 500 genes that are regulated in a miR430 independent fashion. We isolated their 3' UTRs and fragmented them to 100 nucleotide fragments. These inserts were used to generate GFP reporters, and collectively thousands of reporters can be injected into zebrafish embryos in the form of mRNA and assessed for whether or not the insert contains a destabilizing activity. And we do that by looking for depletion of reporters at a later time point. So if a fragment contains a destabilizing element, it will be depleted in a later time point. We can quantify this using Illumina sequencing of the injected reporter constructs, and we can identify regions where reads corresponding to that region are depleted at a later time point, which would be consistent with harboring a destabilizing element. So to visualize this destabilizing activity, we can plot the fold change of reads overlapping each nucleotide across the 3' UTR, and if a specific region is depleted in reads, we can see it as a downward peak. We first wanted to see if this method can be applicable to miR430, so a known destabilizing element. And so in this case, if we take a miR430 target 3' UTR, we know exactly where the destabilizing element is. And the expected result would be depletion of reporters containing miR430 seeds in wild type and stabilization of those reporters when miR430 cannot be transcribed in alpha-amanitin or when miR430 is inhibited. And we find, this is ugly, and we find that we can in fact identify a miR430 seed region. So here's a UTR of a miR430 target, and here you see a peak perfectly centered around the miR430 seed when we indicating there's a depletion of reads in the wild type scenario, and likewise this is true when we look at the miR430 inhibition condition. Furthermore, global analysis of all miR430 regions demonstrates that this method can detect miR430 activity. This is a meta plot showing all regions containing miR430 sites in our experiment, and we can in fact even detect the relative strength of clearance